So in this segment of the lecture, we're going to look at the first three parts of aerobic respiration. And the first three parts are called glycolysis, pyruvate oxidation, and the citric acid cycle. And in this diagram, you see an artist's rendering of the mitochondrion as a section, so cut through so you can see inside. You can see glycolysis. They're putting this little icon for glycolysis, my pointer here, indicating that it's happening outside of the mitochondrion. And for any cell that doesn't have mitochondrion, it's happening in the cytosol. And then for the cells that do have mitochondria, pyruvate oxidation, they're indicating here that it happens inside the mitochondrion, as well as the citric acid cycle. The citric acid cycle goes by another name, which is the Krebs cycle. And it's also even called the TCA cycle occasionally. Your textbook leans towards the citric acid cycle. In my training, I always learn Krebs cycle. They mean the same thing. So these first three parts of cellular respiration happen in the cytosol for glycolysis, and then inside the mitochondria for those cells that have mitochondria, pyruvate oxidation and the citric acid cycle. And then the fourth phase, which I won't cover in this lecture segment, but it'll be in the next one, is the electron transport chain chemiosmosis oxidative phosphorylation. So that's a mouthful. That also happens inside the mitochondria for cells that have that. So as we go through the first three phases, we're going to look at what the pathways are, just very generally. We're going to just summarize each of the first three, and then we will create a chart that summarizes all the reactants and products as well. So the chart will come at the end, but there's some more information in here that you also need. So glycolysis, and you can look here in the diagram, glycolysis happens in the cytosol, is a 10-step biochemical pathway. So what that means is there are 10 different enzymes that work in a sequence. So you'll remember that a biochemical pathway involves a sequence of enzymes, starting with a very starting substrate. In our example, it's going to be glucose. And then that substrate is converted by the first enzyme into an intermediate, which is then converted by the second enzyme into a different intermediate, and so on and so forth, 10 steps. And ultimately, you're going to start with one glucose. So glycolysis converts one glucose into two molecules called pyruvate. Each one glucose contains 6 carbons. It's a 6-carbon sugar. And the two pyruvates each have 3 carbons. So really what's happening in glycolysis is glucose is being broken into 2 pieces. So it's the first part of aerobic respiration. It's not all of it, but it's the first part. And the word glycolysis means splitting sugar in a rough sense. So glyco refers to the carbohydrate concept here. We've got a sugar, a monosaccharide, and lysis is breaking or splitting. So really what we're doing is we're breaking glucose into 2 pieces. And but there are 10 steps. It takes 10 steps to make that happen in such a way that you end with 2 molecules called pyruvate. In this process, there is going to be, directly as a part of this process, there's a production of what we know as 2 ATP. So you remember ATP are the preferred energy currency of the cell. We say there's a net production of 2 NATP molecules. And the reason we say net production is the cell actually uses up 2 ATP initially. We call that the investment, or the energy investment. And that activates some of those early intermediate molecules, and that activation is needed to get things going. But by the end of the process, we're going to get back 4 ATP. So we use 2, but we get 4, and so the net profit or the net production is 2 ATP molecules. And then we also end up with 2 electron carriers. Remember, NAD plus can pick up electrons and become NADH. So there are actually 2 NAD pluses that pick up electrons and become 2 NADH. And those are those electron carriers. So glycolysis is the breaking of glucose into 2 parts we call pyruvate. The net production of 2 ATP and 2 NADH also occur. And all of this happens in the cytosol. Once pyruvate is formed, in this slide, what they're trying to show you is pyruvate is like a decision point in a cell. In other words, if a cell has oxygen and has the means to proceed with aerobic respiration, then it will follow this path here. So this path is with oxygen. But if a cell does not have oxygen, it will follow this path. So they're just depicting it as sort of this decision point, this fork in the road in which whether or not a cell has oxygen available can determine what happens next to pyruvate. So glycolysis is common to all cells, but then the fate of pyruvate after that point depends on the details of what that particular cell has available. And so on this side of the decision tree, sort of the purple or pink side here, these are going to be, what they're showing here really is fermentation, but also what can happen without oxygen is anaerobic respiration. And we will look at those briefly towards the end of one of the coming lecture segments. But in this lecture segment, we're really going to follow this path. If the cell has oxygen and has all the machinery, it will undergo then this path. So pyruvate is formed from glycolysis. Pyruvate then is going to be oxidized to acetyl-CoA, and then acetyl-CoA is going to enter this cycle called the citric acid cycle or Krebs cycle. And so that's the path that we're going to follow right now. So at this point, we have 2 pyruvates that are produced from glycolysis. And the next phase then, when oxygen is present, is called pyruvate oxidation. Pyruvate oxidation is only a one-step process. There's only one enzyme, and the enzyme is called pyruvate dehydrogenase. And pyruvate dehydrogenase is the enzyme that catalyzes the conversion of pyruvate to something called acetyl-CoA. Pyruvate is a 3-carbon molecule, and in the process of pyruvate oxidation, one of those carbons is going to break off. It's a carboxyl group. It's going to break off and be released as carbon dioxide. That's called decarboxylation. The remaining 2 carbons then are called acetyl. So 1 carbon breaks off and is released as a carbon dioxide. The other 2 are still remaining, and the name is now acetyl. Acetyl joins to a molecule called CoA, so we end up with acetyl-CoA or acetyl coenzyme A. But it's not that important. It's still that 2-carbon part of what was pyruvate. And we also make an NADH, an electron carrier, picks up some electrons in this process. So for cells that have a mitochondria, I mentioned before, this happens inside the mitochondrion. But for cells that don't have a mitochondrion, it can still happen inside the cell. And so prokaryotes would be an example of that. They don't have mitochondria. And a little fun fact, arsenic is a poison because it's an inhibitor of the enzyme that catalyzes this step, which is called pyruvate dehydrogenase. So inhibitors bind to an enzyme and stop it from working. And so arsenic is very poisonous because when an inhibitor stops a respiratory enzyme, I think you can imagine how dangerous that is for the cell and the organism. So for each 3-carbon pyruvate that comes into this stage, pyruvate oxidation, your products are going to be 1 carbon dioxide, 1 NADH, and 1 acetyl-CoA. Acetyl-CoA is what goes on to the next phase. But you will notice that at the end of glycolysis, we actually had 2 pyruvates. So for considering it from that point of view, we actually have 2 pyruvates coming into the pyruvate oxidation phase. That makes then 2 CO2s, 2 NADHs, and 2 acetyl-CoAs ultimately for the 2 pyruvates, the 2 3-carbon pyruvates. So we multiply everything by 2 to really get the total yield here. For 2 3-carbon pyruvates, you get 2 CO2s, 2 NADHs, and 2 acetyl-CoAs. So the acetyl-CoA then, and you can see it right here if you look really close, the acetyl-CoA then is a reactant for the next phase, the citric acid cycle. The citric acid cycle is a cycle because, and when we use this word in biochemistry, what it means is that a molecule that is a reactant is going to then be regenerated and end up as also a product. So something in the cycle that you start with, then it reappears at the end, not for magic, but it is regenerated by the end. So you'll see where that happens in this process. But the acetyl-CoA comes in from pyruvate oxidation. This is still happening in the mitochondrion. We're in the matrix of the mitochondrion. And so what the citric acid cycle is going to do is it's going to oxidize the acetyl. And it's a 9-step cycle. So that means there are 9 enzymes in this. And it's a cycle, so you're going to start with something and that you also end with. The molecule that you start with and end with is right here. It's called oxaloacetate. 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 When you're thinking about how to pronounce these, just go very slow and kind of break it up into multiple words. And my advice would be just to emphasize every syllable evenly as you're starting to learn it, and that usually works pretty well if you can't tell where the emphasis might be. But oxaloacetate is already there in the matrix of the mitochondrion. It's a 4-carbon molecule, and it's going to then be covalently joined to the 2-carbon molecule of acetyl. And, that's 4. 2 and 4 makes 6. So the first product of the citric acid cycle is a 6-carbon molecule called citrate. And that's where the name citric acid cycle comes from, because another word for citrate is citric acid. So the first product that's made in this part of respiration is citric acid. So the 2-carbon acetyl that came in from the last phase, pyruvate oxidation, and the oxaloacetate that's already there are joined together to make citrate. And there are, like I said, 9 enzymes, so 8 other enzymes that are going to act on that citrate in steps. And what you can see in each step is what's produced. So from here where the citrate is, you can see in the next step we get carbon dioxide and 1 NADH is made from NAD+. Here we have another step where that intermediate is converted into another intermediate, another CO2, another NADH. You have an ATP that's made directly in the process. You have another electron carrier that picks up electrons. It's called FAD, and that makes it into FADH2. So just like NAD is an electron carrier, FAD is a very similar electron carrier. So it picks up 2 electrons. Another NADH is made over here. And when we get back to here, oxaloacetate is reformed, regenerated. So that's why this is a cycle, because oxaloacetate is not only a reactant, but it's also a product. And so that oxaloacetate can continue and be joined to another acetyl that comes in and go through the whole process again. So let's see a summary of this. So there are 9 enzymes in the citric acid cycle. For each 1 acetyl-CoA coming in, these are your products. 2 CO2s are released, 3 NADHs are made, one FADH2 is made, one ATP is made, oxaloacetate is regenerated, which makes why this is called a cycle. And so that is what you produce just from one acetyl coming into the citric acid cycle. However, we have two acetyls coming in, so we have to, just like before, we really have to multiply all these things by 2. So when we multiply all these things by 2, we see the real total yield of the citric acid cycle or the Krebs cycle. All right. So for every 2 acetyl coming in, then we double everything. All right, so let's summarize at least the reactants and products of these processes, and I'll probably add into it also. I'm going to add oops. Let's see if I can get, okay, discard. All right, I'm going to type in here. And you are going to be able to see this. All right. So the name of the stage, the first stage is glycolysis. This has 10 enzymes. So it's a 10-step process. The location is usually said cytoplasm or cytosol. At least cytosol is a little better. And then the reactants for glycolysis, I'm going to need to make a smaller font here. The reactants for glycolysis, we start with 1 glucose, and then we're going to end with 2 pyruvate. Also, we're going to start with 2 NAD+, and over here we're going to generate 2 NADH. We're going to, I think we'll just do the net on the ATP. We'll start with 2 ADP and 2 PI, and we will net 2 ATP. Although I will ask you later about the investment. Remember, there's an investment of 2 ATP, a yield of 4, so the net production is 2, the net profit. All right, and so that's it. So 1 glucose, 2 NADH, 2 NAD plus, and 2 ADP plus 2 PI yields 2 pyruvate, 2 NADH, and 2 ATP. And that's just for glycolysis, those 10 steps. Then you have pyruvate oxidation. So the pyruvate that's generated from the first stage, it becomes a reactant for the second stage. For cells that have a mitochondrion, then this happens in the matrix. For cells that don't, it can happen in the cytosol, depending on if they have the oxidation. And what we have over here, remember, this is just one step. I'll put that over here, one enzyme. It's called pyruvate dehydrogenase. And this one converts for every, we're going to go with 2 pyruvate. You're going to get 2 acetyl-CoA and 2 CO2. And on here, I'll put the 2-CoA over here. And then, as far as NAD, you're going to get NAD, 2 NADs are converted to 2 NADH, meaning electron carriers. And there is no ATP made in this phase. So one thing you might note is that glycolysis doesn't make any CO2, and pyruvate oxidation doesn't make any ATP. But they both make 2 NADH. And they both do some oxidation of the organic compound. And then the last one that we've just talked about is the citric acid cycle, or the Krebs cycle. This one is 9 enzymes. It's a cycle. The location, we're still in the mitochondrial matrix for the cells that have that. And then for reactants and products, we're going to take the 2-acetyl-CoAs that I'm just going to say 2-acetyl. Nobody worries too much about CoA. 2 acetyls that we started with, and we're going to generate 4 CO2s from those. Wow, that's even smaller. Hang on. How small can this get? All right. And then here, we're going to get 6 NAD pluses become 6 NADH. And then we have 2 FAD becomes 2 FADH2. And then 2 ADP plus 2 PI yields, oops, that's the wrong side, sorry. And why is that going so small? That yields 2 ATPs. Oh, I don't know why it's doing that. 2 ATPs. Okay. So those are reactants and products of the citric acid cycle. And then if you want to put, actually, let's go ahead and put 2-oxaloacetate on both sides. Oxaloacetate. And then 2-oxaloacetate. On this side, that's what makes it a cycle. The only rule for a cycle is that something you start with has to be a product as well. Not everything, but something. So oxaloacetate makes this a cycle. All right. So what we have here is, if we look at our products and we kind of group things together, then what we end up with is our products up to this point. If we look on that chart, we have made a total of 6 CO2s because we made 2 in pyruvate oxidation and we made 4 in the citric acid cycle. We've made a total of 4 ATP directly produced, 2 of them in glycolysis, 2 of them in the citric acid cycle. And then we have piled up NADH and FADH2 from all 3 of the phases. At this point, let's talk about where these molecules go from here. CO2, when it's made, it can diffuse out of the cell right away. It doesn't hang around. As soon as CO2 is made, it diffuses across the plasma membrane and out of the cell. The ATP that are made directly in this part of the cell of respiration can be used right away. So these are probably used, although we're going to count them in our total, but they're going to be used pretty quickly. It's like cash in your pocket. You spend it right away. So we're going to count this later, but probably when it first is made, it gets used right away. But then it's these guys here that we're sort of left with when we think about the 4th part of cellular respiration. So the NADH and the FADH2 that we have made during the first 3 phases of respiration are the things that are going to go on to the 4th stage. And the 4th stage, if I can spell correctly, is called electron transport chain chemiosmosis oxidative phosphorylation. So it has this very long name. And so that is where we're going to start in the next lecture segment. I'm going to go through that 4th stage, which is going to be where we get most of our ATP, because at this point we've only made 4, and I told you we should be making a total of 30 something. So the biggest yield of ATP is actually going to come in this 4th stage of respiration.